Well, good morning, everybody, or good afternoon, depending on where you are. My name is Arthur Brandwood. I'm the founder and principal consultant of Brandwood Biomedical, and I'm here to present to you our July 2018 live webinar on design verification of 3D printed devices, biocompatibility, sterility, and biomechanics. What do I mean by that? What we're going to look at today is how you tackle the problem of verifying and controlling the design of devices where every single one is different. This challenges the normal paradigms of design control and process control in medical device or any other kind of manufacturing, where you're making one-offs each time and you can't rely on testing on a standard item. But before all of that, I'd just like to make a couple of housekeeping. Firstly, this is a live webinar. At the end, we will do a question and answer. You will find on your little control bar, you should be seeing a black control bar. There's a Q&A button. You can click there and you can send a question to me at any time. I'll receive all the questions, and at the end of the webinar, we'll do the Q&A. A little bit about Brandwood Biomedical. And if I can, there we go. We are a regulatory technical advisory firm covering comprehensive services in regulatory affairs, quality assurance, and reimbursement. We do everything in the entire life cycle, from initial strategy to regulatory submissions through to post-market compliance and surveillance. We can handle reimbursement, clinical trials, quality management systems, either building them or auditing them or looking after your vendors. And we can hold licenses and act as an authorized representative here in Australia, in China, in Taiwan, and in a number of other markets. We are headquartered here in Sydney. We have our own offices in New Zealand, in China, in Hong Kong, in Taiwan, and partners and partner offices across the rest of the world. That's enough about Brandwood Biomedical. Let's just talk about 3D printing. This device made the news in Australia a few months ago. This is 3D printed sternum and ribs, printed from 3D printed metal here in Australia, implanted into a patient in Europe. How do you deal with something like this? It's a complicated device. It's unique. There's only one like it, and it was made for that particular patient. And so what I want to talk about today is how you go about developing devices and manufacturing devices like this, where you are making each one different, and how you assure that you are meeting the design requirements for these devices. So here's our agenda. I'm going to look a little bit, just refresher on what design control and design verification is, just to remind everybody what we're speaking about. And then I'm going to talk a little bit about process verification and validation as well, and what are special processes and how custom devices really bring that whole idea of special processes right through the whole design. And then we'll talk about some specific cases, specific examples, and we're going to particularly look at biocompatibility, at biomechanics, and sterility, and then maybe finish with a few strategies. So many of you will be familiar with this diagram, the so-called waterfall diagram. This particular version, drawn by an anonymous bureaucrat in Health Canada several decades ago, and has been recycled through guidances and websites and webinars, and it's the one document, it's the one graphic that appears everywhere. The whole point about the design control process is it starts off with the user. You go about developing a medical device based on the inputs from that user. You end up with a final device, which you then do two things. You test it in the laboratory to see that it actually meets those user needs, and then when you're finished with that, you then test it in the clinic to see that it works in the real world. It actually does work in the hands of the user. That's all very conventional. But when you have a 3D printed, custom-made device, and really we're talking here about custom-made, not 3D printed, mass-produced, design control has to be looked at 2 levels. You can define high-level user needs that are the same for all custom devices in a series. So if a laboratory is turning out craniofacial implants, each one is of a different design to meet a specific patient, those implants all need to be sterile. They all need to be made of biocompatible materials. And they also all need to have a mechanical design that's going to be safe. They're going to need to be strong enough to do the job. The first 2 of those, the problem is the same for every device in the series. If a laboratory turns out 100 of them over a year, they still need to be biocompatible. They still need to be sterile. But the mechanical design is different for each one because they are each one individually manufactured to fit an individual patient. And so there you have a somewhat different challenge in validating for the individual user. So we can do verification at 2 levels. We can verify that something is sterile, and we can do that in terms of all devices. But verifying the mechanical function of an individual device when each one is different, a different shape, different design, that's done at the device level. So how do we manage that? Well, if you are, as Charlie Chaplin was doing, making something that's the same every time, passing down a production line, then there are tried and tested ways of doing this. You can measure quality control, see if he's tightened the nuts appropriately on the device he's building there, make measurements and develop a control chart like I see here, where you make time series measurements and make sure that you keep close to your target average in the middle, the dash line in the middle, and within the limits, the upper and lower limits that you set that allow production variability within those limits. That's all very conventional. That's how standard quality control is done. And you can develop statistics around that and monitor the entire process quite easily. But there are a number of processes where you can't do that. There's the concept of a special process, a production process which requires validation, and that is simply because you cannot make a measurement on the product without destroying the product. Examples of special processes, sterilization being one of them. You can only determine if an individual product is sterile by opening the packet and putting the product in a broth and seeing if you can grow anything, and that's then destroyed the product. Aseptic filling, the same. Sterile package sealing, you can only determine whether the package is properly sealed by attempting to open the seal. And treatments such as gluing, soldering, welding, or additive manufacturing, building up layers, you can only tell if that's been done properly by mechanically testing the device which is going to destroy it. So these are what is known as special processes, which require validation by means of things like doing a production run of a defined series of devices and then taking a statistical sample from those, or taking a whole, testing the whole lot, doing 100% sample from a test run, from a validation run, to see that if you control the conditions in a defined way, that you get an understood and defined output which pre-validates the production. There are very well tested ways of doing that with things like sterilization, where you can do known cycles and half cycles on product and measure the sterility in a statistical way. The Bible on this, for those that think that GHTF is long ago, the Bible on this was written by the Global Harmonization Task Force with their guidance on quality management systems process validation guidance. That's document SG3N99, the number 99 document that came out of the GHTF, and it's still the best written guidance on process validation. So just as an aside, that's process validation. But really, when you have custom devices, you have to bring the same principles into the design phase. So you need to be thinking about, okay, how can we apply generic principles to a product design where we can't actually test the product without breaking it? So if the device is an additive manufactured device, the examples we gave at the beginning that we're going to focus on in this webinar, it needs to be clean and sterile, it needs to be strong enough, and it needs to be biocompatible. But every one of these devices is different, and so every one is an essentially special in terms of a special process in its manufacture. So let's deal with these one at a time. Firstly, sterility. Now, I've worked with a number of 3D printing custom device manufacturers who say, well, we'll make the device, we'll make it clean, but we'll then supply it to the clinic, and the clinic can worry about sterilizing it. They've got autoclaves, they've got ethylene oxide sterilizers. But regulations in just about every market actually say that if the manufacturer provides a device which is intended to be sterilized by the end user in their facility, then the manufacturer is obliged to provide in the instructions for that device a sterilizing process which the manufacturer has validated and holds evidence that that process works. So whether you're going to sterilize at the clinic or whether you're going to sterilize it yourself in your own manufacturing facility, you must have a validated sterilization process and cleanliness as well. So this also extends to things like endotoxin testing. Now, actually, whether you've got 3D printed customized devices or whether you've got mass-produced devices, all the same, in fact, you can take the same approach to sterility validation. So sterility validation is usually done by taking worst cases. So a good example being if you're making, say, procedure packs, where you might have a number of medical devices within one package, which is all sealed up within a Tyvek pouch and then sterilized. The way to do that is to test the worst case. So do a sterility validation on a series of devices which are worst case. And in the context of a additive manufactured implant device, you may wish to then choose devices which are the largest surface area, for example. The most complex shapes may be devices which have cavities or porosity, where it's more difficult for a sterilant like ethylene oxide to penetrate. So you'll need to do is to justify what is the most complex and difficult device that you are likely to ever manufacture, and then test that. So you would like to, the way to do it would be then to actually build a series of devices which are effectively worst case devices, and then you can run those through your production process and run them through the package them as they would normally be, and then run them through a sterilization validation. And I'm not going to talk about how you do sterilization validations. That's not what we're really here to do today in the general sense. But there are well-defined ways of validating sterility, all written down in ISO standards. So the ISO 11137 series provides means of sterility validation for irradiation, for gamma sterilization. ISO 11135 provides sterility validation methods for ethylene oxide, and ISO 17665 series provide moist heat sterilization, in other words, autoclaving. And there are a number of others for different types of sterilization. There's a whole body of standards. So the way to approach this here for custom devices is to understand that you are going to have a range of different geometries, a range of different sizes, possibly different porosities or different cavities in the device. Figure out what's the most difficult thing you're likely to make, what's the most complex shape, make that, and test the devices made like that. You can use your 3D printer to make a whole series of them the same and test those in a conventional sterility validation program. But biomechanics is different. Biomechanics, every device is different. You can go to the internet and look and search on 3D printed devices or additive manufactured devices, and you'll find dozens and dozens of pictures that look like this. This one happens to be, and I acknowledge it comes from Kilinium, from their website, from some of the devices that manufacturer makes. And the point is that every 3D printed implant is different. The technique is widely used for craniofacial implants. It's used for dental devices, for things like mandibular splints. It's used for the sternum that we saw at the beginning of the presentation. Everything that you make is different, which means that the mechanical design is different. The loads transmitted through these devices are different. So how do you know that the device is going to be strong enough, tough enough, and of the right stiffness to do the job? And particularly since most of the applications at the moment are replacing bone. Now, the argument is often, well, the skull is not a load-bearing bone. It's not like a major long bone, it's not a joint, it's not load-bearing. Maybe so, and we'll come back to that to the moment, but the point is what you're going to need to think about is how you do the evaluation here. Now, you can consider a direct evaluation. You can establish the limit cases in the same way that we were thinking about what's the most difficult device to sterilize. You can think about, well, what's the most challenging mechanical design? What's the likely thinnest or thickest or narrowest or most extreme aspect ratio? What's the highest porosity? What is the maximum size of device that we might make? And think about that in terms of mechanical design and say, well, okay, can we define a set of parameters, which is a sort of design envelope? We will not make anything that's thinner than certain thickness. We will not make anything that's got an aspect ratio above a certain amount. We will not allow a part to be narrower than a certain amount. We will control our porosity so it sits within a particular range. We won't make a device that's bigger than X. You can do that sort of thing. The other approach is maybe to compare your material to a predicate material and say, well, we've got a material here which is as stiff and as strong and as tough as another material which is used in this application and for which there's a clinical track record, and make the comparison that our material is the same as that one over there. It has a record of safe use, and therefore, by comparison to the predicate, we can claim that the new 3D printed material is going to perform in the same way. So you can see the sort of things there. But there are some hazards here. What's elephants and rhinoceros got to do with this? Interpretation of loading in bone is complex. I, many years ago, started my career in zoology. I don't know how I ended up in medical devices. That's a different story. But one of the first things I learned as a graduate student was that rhinoceros is the largest animal that gallops, and an elephant can't gallop because an elephant gets to the point where it is so big that for structural engineering reasons it would break its femur if it tried to gallop. It's too big and heavy. So there are other things going on in design of bone, and elephants and mice is the next example. The point here is that if you look at the stresses within a bone, which is what really matters. It's not the absolute forces, but it is the stress that's applied to the bone, and stress is force divided by surface area. So whether it's compressive stress or tensile stress or a bending moment or a shear stress, it's to do with force and surface area. And there's this thing called Wolff's Law, which says that bone adapts according to a load placed on it, which means that for the mouse, I think that's a rat actually, but for the rat and the femur of a rat, and for the elephant has a much thicker femur than the rat does, but when you work out the forces generated by the dynamic loading on a rat as it runs around compared to that of an elephant as it ambles along, the stresses turn out to be the same. In other words, the bone remodels so that the stresses through the bone are roughly the same. And that's what's going on in the skull. So it may not be taking large loads and bearing the body weight, but it's still got to be designed to withstand impact stresses. It's also got to be designed to withstand the loads generated by the muscles around the skull. And in fact, some of the jaw muscles are quite powerful and place quite large loads on those. So there's no such thing as a non-load bearing bone, is my point. And so you do need to think about, well, what are the actual stresses on the bone, even though you might think it's not load bearing. Now, 3D printing comes about because of software, because of what we can do with medical images and how we can take those medical images and create data files that we can then feed into additive manufacturing equipment and generate a medical device which fits into the patient anatomy. So that actually provides another opportunity. So when we're looking at biomechanics, you can do this sort of thing, and this is a publication where the authors have looked at best and worst cases of particular designs of craniofacial implants and done finite element modeling and looked at the stress intensities. So here's another way of perhaps validating a device where you could, for example, plug each model into an FEA, each device that you make, and systematize this so that you verify that in a finite element model that you are not exceeding particular stress intensities, that your design doesn't have stress raisers, narrow parts on it or whatever, that are going to raise the stresses to the point where you might be jeopardizing the integrity of the implant. So take advantage of the computerization because that might actually allow you a straightforward way of building what are relatively complex models, but coming out of the existing software infrastructure that you have for handling the data sets. Again, I said before, you could simply go to look at a comparison with an established predicate. Here's a surgical mesh, not 3D printed, produced by another means, but certainly there are manufacturers producing mesh-type products now, which are 3D printed, and you could compare them to established devices and look at the mechanical properties and take that particular approach. And what about standards? So there's a surprising amount of work being done. I haven't particularly the ASTM, the American Society for Testing and Materials, has a whole working group on additive manufacturing standards. And this is a graphic from their website which talks about the structure of the additive manufacturing standards that are being developed by ASTM and others. So we have, for example, a bunch of standards on terminology, a whole bunch of standards on processes and materials and qualification certification methods for materials, requirements for purchased additive manufacturing parts, and non-destructive evaluation methods. And that's particularly important, non-destructive evaluation methods that do allow you to evaluate the material in a device without destroying it. Test methods, how to develop test artifacts, general test methods, performance, and then a whole bunch of standards around design and data formats, data structures and metrics. So that leads on to a bunch of more specific standards on materials, on processing, on doing testing, round-robin testing, comparing different laboratories, that sort of thing, when you're developing processes. So a lot of work going on. Much of this in draft at the moment, but a lot of work going on, and of course not just applied to medical devices. So this particular group at ASTM is working across aerospace, medical, automotive, and a whole range of industries. But it's coming. The standard infrastructure is on the way. So turning to the last example, I want to talk a bit about biocompatibility. Now, if you are developing a hip joint like this one, we have a stem, we have a ball, we have a cup, and we have the insert for the cup, the polyethylene insert, all made out of conventional materials which are very well established. So this device might be made of one or more, usually one metal alloy that for all the metal components, and then the polyethylene insert. And if you go to the standards that describe these surgical alloys, ASTM standards for things like surgical titanium or for cobalt chrome, will all contain a passage that says these devices, if they are appropriately clean and appropriately processed, are considered to be biocompatible. These materials have an established record of biocompatibility, and you don't need to go off and yet again test surgical steel. You don't need to go off and test yet again titanium vanadium alloy. You shouldn't be needing to go off and test nitinol, providing you can show that your manufacturing process doesn't add anything else, that you don't contaminate the metal in the processing. That's fine. So these are mass-produced devices produced by forging, machining, whatever, and the materials are very well understood. But the starting materials for additive manufacturing look like this. This is just a picture of some of the metal powders that might be used in additive manufacturing, or you might be using polymers in a polymeric device, reels of polymer that feed into a 3D printer. The question then is not so much what's the composition of those little piles of powder, but what's the composition of what comes out at the end. And you can source titanium powder, which is claimed by the manufacturers that once it's been put through a defined additive manufacturing process, complies with the ASTM standard for titanium surgical alloy. Is that true? Well, the only way you can do that is for you to measure it afterwards and do the metallurgical analysis to show that in fact that's what you've got. And in fact, most material suppliers will tell you that they've designed their material in a particular way for a particular application, but it is incumbent upon the device manufacturer to verify that what they get out of the end actually does meet requirements, and it's not the material supplier's job. So how are you going to deal with that in terms of biocompatibility specifically? So most people look at biocompatibility, look at the ISO 10993 standard series, and here's the list of what's going on in ISO 10993. The reason for the color coding here, this is just a slide that shows some of the particular parts that have recently been amended or updated. And it's a living set of documents, taken about 25 years to develop all of this. We're not going to get into how you do biocompatibility in detail. It's not about the generic question there. It's how do you apply this to additive manufactured devices. So really, what you want to be doing here is using materials that are understood and qualifying those materials in the form of a final device. So the evaluation needs to be applied to the finished device. So that is the device which has been processed, where the starting materials have been turned into a printed part. That part has been subject to post-processing. It may have been polished, it may have been cleaned, it may have had further machining steps. It has then been packaged and then sterilized. And it's that bit, the device that's been through all of that, that actually needs to be evaluated for biocompatibility. Because what you want to know is not just that the materials are safe, but that you haven't added anything to them, that you haven't got sterilant residues present, that you haven't got lubricants and processing aids from the machining steps or from the cleaning steps. You haven't got cleaning residues, cleaning aids residues left in the device, all of which could be toxic substances. Now, the good thing about the ISO 10993 biocompatibility standards is that these days there is a considerably greater emphasis on chemistry than on immediate resort to biological testing. The various animal tests and cell culture tests and so on that are done in biocompatibility testing are expensive, they're slow, and difficult to interpret at times. But if you can do good chemistry, if you can characterize the device by doing a chemical characterization, you may be able to show that you've got nothing to worry about. And particularly if you're able to show that your 3D printed metal device, for example, actually is chemically and physically equivalent to a mass-produced surgical alloy device. So So really, it begins with understanding the materials, and I point there particularly to the boxes on the right where the ISO standard 10993 has parts 9, 13, 14, and 15, which are all about identifying and quantifying degradation products and materials. And part 18 is about chemical characterization of materials, and part 19 about physicochemical, morphological, and topographical characterization. And then part 17 provides a framework for risk assessment where you can quantify what might come out of a device in terms of leachable substances, residues that can be dissolved out of the device and end up in the tissues. And if you can characterize those, identify what's present by doing extractions and analytical chemistry, then you can make some reasonable judgments about whether there is a hazard that arises from the substances that may come out of the device. And for 3D printed metal devices, there shouldn't be anything coming out at all, or very little. There may be some low-level release of metallic ions and corrosion products, but if they are 3D printed surgical steels, 3D printed surgical titanium alloys, then you would not expect there to be much release at all. And then if you can show that through chemical characterization testing, you can make an argument that you do not need to proceed to biological testing because, in fact, there's no release. So looking at that, the question is, are we sure of the composition of that final material? Is it really the same as, for example, a mass-produced ASTM surgical metal, which might be forged? And so how do we do that? Well, manufacture some test samples, use your additive manufacturing to make test samples. The beauty here, of course, is the ISO standards call for particular sample configurations because most of this work is done by doing an extract. And the Part 12 standard in ISO defines the volume to surface area ratio of the test sample that you immerse in the test solvents to do an extract. So 3D printing is great because you could actually print test samples that provide you with the right volume surface area ratio to make it easy. So you can print your test coupons using your additive manufacturing machine. Manufacture the test sample, meet those surface to volume ratio requirements, and then analyze it. Do metallurgy if necessary to look at the final structure of the metal, and do exhaustive extractions and chemical characterizations. An exhaustive extraction is when you take the sample and you place it into a solvent for a defined time period, usually at an elevated temperature. Typically for implants, you'll be looking at 70 degrees for 24 or 70, 48 or 72 hours, but an accelerated temperature for a reasonable length of time. And then you will take the solvent residue, you'll analyze it and see what's in there, and then you'll do it again. And you keep doing that until the latest step has produced a residue which is less than 10% of the total. In other words, each cycle you do this, you'll get less out, and when the last step is less than 10% of the total, then you consider to be there. You've extracted more than 90% of what's extractable. For most metal parts, you would expect that to be on the second cycle because you wouldn't get much out. But for a polymer, you might go longer. So if you're making a methacrylate additive manufactured device, you might find that you've got residues in there of unreacted monomers and other things, and you might be doing a longer extraction. And then what you could do is do the same thing with conventional off-the-shelf machined materials which are equivalent. So you can do two things here. You can do an exhaustive extraction, look at what chemistry is present in your additive manufactured device, and on first principles say, well, we can characterize what's there, and it's all very low levels, and it's not hazardous. You could take this one step further and say, well, here is an equivalent device manufactured not from additive manufacturing processes, but from conventional processes, which has got an established track record, and you can do a comparative analysis of that and show that what you get coming out of your device is similar to or less than what comes out of a conventionally manufactured device. So there's that way to look at it as well. So the idea is to demonstrate biological safety by chemical characterization. If you can't do that, if you do find you've got a fair amount of stuff coming out, if this is a new material and you've got substances leaching out which are dissolving out of the material, which may or may not be hazardous, then you may need to proceed to actual biological testing, starting with cell culture, cytotoxicity, and then going on through irritation, sensitization, systemic toxicity, and all the genotoxicity tests, depending on the application of the device. And the more invasive, the more testing you've got to do. And don't forget to do all of this testing on product which has been through the cleaning and sterilizing steps. So often manufacturers make the mistake, particularly when coming to this for the first time, of testing samples which have been straight off the printer, but not been cleaned and not been sterilized, and the testing on those non-clean, non-sterilized samples is not valid because it doesn't represent what may be added to the device in the cleaning and sterilizing steps, or what may be taken away in the cleaning step. So in summary, the challenge with 3D printed custom devices is that each one is different. You cannot simply test a type. You don't have a mass-produced device where everyone is the same, where you can test that and validate and verify the performance by testing a type, essentially. So what we need to do is effectively establish limit cases. Look for the physical configuration, size, shape, porosity, cavities, etc., that makes it the device most difficult to sterilize, and test that. Make that as your test case and run that through a sterility validation programs, because that will validate all of the less difficult ones as well. Look at the most mechanically demanding geometries when you're thinking about the mechanical integrity of your device, and validate that. And then for biocompatibility, you are looking to demonstrate that you have non-toxic finished devices, that the materials in the finished form are non-toxic, preferably by chemical characterization, but possibly also by biological evaluation. So we'll stop there. We've been going for a little over half an hour, and it's now time for some questions and answers. I remind you, you've got a little Q&A box. If you've got a question, just simply type it in for me, and we'll see what we can do. And just looking at here, I've got a question here from Alfredo, who asks, what about using simulation, i.e. CFD, as a means to provide data on custom devices? Alfredo, we've got a lot of acronyms here. The MDIC in the US, I know what US stands for, has an initiative around data from a virtual patient being applied instead of traditional clinical trials. Could this not be extended to custom devices by way of simulations? And I think you're right, and you supplement that by saying you're thinking more about finite element analysis, and that's something I did touch on, is that the whole point here is that custom-made devices using additive manufacturing processes, the whole process is built upon software models of what it is you want to build. So based around imaging of the patient, passing that data set through a set of software that end up coming up as an instruction file for an additive manufacturing instrument or machine to produce the hardware device. And yes, I think it's clearly going to be amenable to FEA. There is an FDA guidance on this, by the way, fairly recently produced on additive manufacturing. It's quite a thoughtful guidance, which talks about some of the things that you might want to consider when doing this. And one of the things it gets into, Alfredo, is it talks about how you control the integrity of the data as you transform it through the different software steps. Because inevitably you'll be introducing error and noise at each step. And so it talks about, well, how can you do that, and how can you then be sure that the final image that comes out, the final model, still faithfully represents the patient anatomy, for example. And how could you also be sure that if you take something from that and feed it into an FEA analysis program, that you actually are analyzing something that's representative of the final hardware. I have another question here from Chris, who says, we are trying to validate an automated 3D printing process that's now replacing a previous manual process in a dental laboratory. And how do we deal with that? And that's a very good question, Chris, because what you've got there is you don't have a mass-produced, predicate device, if you like, which is produced using conventional machining or molding processes, but you're actually making things by hand. So really, I think what you're needing to do there is to head off and look for other predicates and look for other devices made in the same way with similar materials. Dental laboratories are typically working with things like dental acrylics, and there are plenty of devices around that are mass-produced using those materials. I have another question here from Stephen. Stephen wants to talk about sterilization and asks, can we just expand a bit on how you might validate the sterilization using a worst case and how that works. So just want to go back and touch on that again. Okay, so sterility validation, typically the way it's done, and I'm not an expert microbiologist by any means, but the way sterility validation tends to be done is that you will take your worst case sample. In here, we're talking about maybe the largest, most porous, most convoluted sample that you might ever make. And you'll take a number of those and you will deliberately, you'll run them through a set of sterilization cycles in the sterilization instrument, whether it be an autoclave or an ethylene oxide device, for example, and you will do a full sterilization, and then you'll do something like a half cycle. In other words, you'll only apply half of the sterilant or lower temperatures to the device, so that you'll deliberately do an inadequate sterilization, and then you can then do a statistical analysis where you look at the number of bacteria that survive the half cycle compared to the number of bacteria that survive the full cycle, and determine sterility assurance from that process. That's rather briefly defined, but that's how you're doing it. Is you're taking a range of devices, a sample of devices which are worst case, and you're passing them through sterilization processes which are both complete and which are less than would be complete, to try to get a measurement of how effective the whole process is. I have another question just come in from Alicia, which says, how can we justify that we have constructed the most complex form plausible? In the case of a maxillofacial, where would the line be drawn? That's a very difficult question to answer, of course. What you need to be thinking of is what are the, if you like, the abstract parameters that you can use to define your device. Maxillofacial devices do tend to be complex shapes. So, but try to think not about the complexity of the overall shape, but what matters, Alicia. So, what, in terms of mechanics, what matters would probably be how thick is the wall, how faithfully does it represent the normal anatomy? Because where you find thicker bone, you'd need to have correspondingly thicker replacement material, at least you would expect. If it's a porous material, think about the porosity, because porosity is going to mean more difficult to sterilize because there's possibility for bacteria to get in amongst the pores. It's also going to mean a different approach to biocompatibility, because porous materials are more likely, with a much larger surface area, are more likely to elute soluble substances from them. And so think about porosity, think about physical dimensions. And when you're thinking about mechanical strength, long aspect ratios, things that are long and thin, are going to be more challenged in a stress, by a stress, than things which are short and fat. Simply simple mechanical engineering. So I think that's how you do it. You have to do it in terms of specific parameters which measure the device, rather than trying to characterize the entire shape of each maxillofacial implant. I have another question here from Jenny, who has asked, who says, we've discussed things like biocompatibility, sterilization testing, and so on. What test is the most different for additive manufacturing than for traditional subtractive manufacturing? So where do we depart most? Well, I've tried to choose some examples here. So for example, sterilization, I hope the point has got across that actually sterility validation is the same for additive devices as it is for any device. You can take a device manufactured by whatever means you like. You simply have to take the worst case. So you might have a set of procedure kits that have a range of different instruments present, syringes and scissors and scalpels and bandages and whatnot, and you choose the worst case out of all of those. And you validate that and say, well, anything I put in my procedure kit we know is going to be sterile. You can have a complicated shape of a catheter, which is made by conventional subtractive, and you take exactly the same approach. So that's not really different for additive manufactured devices, except that you just need to look at the configuration that you might manufacture. And if it's a complex craniofacial implant, you look at the most complex shape. I think the thing that's really the most different is going to be biocompatibility. And that is because there is such a big transformation in the materials during the manufacturing process. When you're making a monoblock cobalt chrome hip, you are typically forging it and then maybe machining it. You are simply taking a lump of metal, squishing it into a shape in a forge, and then when you've done that, you might machine it to the finished form. You're just taking bits of metal off. So you're not really changing the material at all when you do that. The only thing you're doing is you're doing a washing step where you will be making sure you've not left any grease residues on the surface. And when it comes out of that, you should just have a lump of cobalt chrome. But when you are doing 3D printing with powders and sintering those, the metallurgy is much more complex. The materials may be more porous, and you don't really know exactly what's in there. So that's going to be more complicated, and that's why it's really going to be necessary to make sure that the finished product in an additive manufacturing process doesn't contain things that you didn't expect. You cannot assume that that metal is going to be identical to a solid piece of surgical alloy that you've simply machined into shape. So I think that's where you're going to get the most difficulties. So let me just see. I think that's the last of the questions. We're just coming up to short of the hour, so we will draw the webinar to a close. Thank you for your attention and for your questions. We do these webinars once every month, typically around the third week of the month. Look out for our newsletter, first Tuesday. No prizes for guessing when that comes out in the month, first Tuesday of every month. And that will tell you what's in the next webinar and provide you with the sign-up details. You've registered for this one, so we are recording this webinar. We'll make the recording available to you, and we'll also provide you with a copy of the slides. So look out for those emails arriving in your inbox, possibly later today or certainly fairly shortly. You can follow us through our website, follow me on Twitter, LinkedIn, and you'll also see these webinars on our YouTube channel, on the Brandwood Biomedical YouTube channel. So it leaves me to say thank you for your attention, and hope you found this webinar valuable, and we'd appreciate any feedback on the content. And goodbye for now.